So I think the reason why we want to kind of like start slow is we did ask everyone that registered for this specific workshop what their experiences was and what they're like wanted to learn, and there was like a really big variability. Some of you had almost touched upon every single technologies, and others had not had any previous experience. So I just want to give you a quick idea about what are the different spatial omics technologies and the different applications you can do. So as a very simple example, here is a spatial transcriptomic data set from a mouse brain, which you see on the left, and this is an H&E stain. So it's hematoxylin, which stains the nuclei, and eosin, which stains the extracellular matrix. For a very long time, this has been the cornerstone of pathology, and it really helps you to study morphology and to assess disease status. However, there is so much more information that is encoded in the cells that are part of the mouse brain. And so what is available? There is like literally like this RNA, protein, metabolites, and how can we capture that? So in order to capture that, we need some kind of like spatial extraction tool. And here I'm giving you the example of, I think this is the PCM CytAssist, where you have specific barcodes and you can extract that type of information from localized spots, so spatially barcoded spots. And so in this specific case, you can identify where each gene is being expressed. And here I'm giving you an example for CALM2. But if you then do it for almost all genes, you can start getting some more like intricate spatial insight about how a tissue is organized, where the cells are, what the cells are expressing, how they're communicating, and how the tissue is organized in general. All right, so what makes a spatial omics technology? So usually it's a combination of one or more of these, like it's sequencing, like I showed before, they can be spatial sequencing based. There's an image almost all the time, but sometimes the technology itself is imaging based and not sequencing based. And then there is also like a level of spatial data science where you have to generate the data, but also like how do you interpret the data? And people have been doing this already for a very long time in different fields, including in geology and ecology and whatever else. And recently, about a year or five now, we are doing this more and more in the emerging field of spatial biology. So what are some of the biological questions we're trying to answer? And my lab really approaches this from the cancer biology field. So I'm just going to give you kind of like a very short example of what we can do. But usually it's about the questions that really require us to understand cellular behavior, not within the cell itself, but within their normal tissue or environment. Like how do cancer cells, how do they grow in 3D? How do they interact with the different other cell types of the tumor microenvironment? These can be immune cells, these can be like your stromal cells, endothelial cells. How do they signal between them? How do they crosstalk? And so to put that historically in a perspective, what we used to do, and here you see as an example, a breast cancer tumor, which is one of our, I wouldn't say favorite, but the most used tissues in our lab. They would take it out, they would cut it, and they would look at it morphology-wise. Pathology would look at H&E, but as I said before, there is so much more at the molecular level. So which genes drive treatment resistance? Is the immune system activated or suppressed? Are they biomarkers that we can potentially find and that can help us to predict response to treatment? So there are so many questions that we can answer at the molecular level. So historically, we just took the whole tumor, we isolated RNA or sometimes DNA, depending what you do, or protein. And that has been really helpful. I think a lot of progress has been made already for like the last couple of decades in understanding like the RNA profile, the larger transcriptomic profiles. But there are certain limitations in RNA-seq, right? And the simplest way of visualizing this is you would throw the whole tumor, you would blend it, and there's like you lose the idea about the individual tumor cells and the individual non-tumor cells that are part of the tumor microenvironment. So then people started doing single-cell RNA-seq, and that has been like really popular. And with single-cell RNA-seq, you kind of like deconvolve that in this individual constituents of your tumor, in our case, or any other tissue you're interested in. And then you can cluster the individual cell types. So now you get information at the individual cell. You can even like do cell trajectories. But this is a workshop for spatial, and so I'm going to tell you why spatial matters. So there's a lot of information at the spatial level that is kind of like hidden if you just dissociate your tumor and then you just have single cells, but you lose the context of the bigger tumor microenvironment. So ideally, we would like to have single cells, but we would like to know where were they coming from, so we have an idea like how aggressive or non-aggressive that specific tumor is. And this is why people have been developing a lot of different spatial omics technologies. But spatial omics technologies have been it's a really emerging field and a very fast growing field. And it's very hard to understand what you need in which situation. And sometimes you need a little bit of both, or you need multiple of these technologies to answer all the questions you might have. So what are the different steps in spatial omics research? There's data generation itself, but then there's the data pre-processing steps, there's the analysis, and finally, if you do all of that, you can do some really complex use cases, which I will show you at the end of this like small presentation. Data generation, so there's definitely a variety in data characteristics. Each technology is very different, and the variety of data outputs, like as you will see in the next slide. So there's a very long history of spatial technology development that originated already many decades ago, and there are different type of spatial technologies, and this is already very outdated, so I should probably update it really soon. I do want to highlight three different technologies because I think they represent a large part of the general different technology that are available. One of them is the sequential IF, seqFISH Plus, and VCM10X. And the reason why I want to highlight them is because I think they are a really good and easy way of explaining these three different big pillars of spatial technology development. You have spatial proteomics here on the left side. In the middle, you have spatial transcriptomics that is based on fluorescence in situ hybridization. And then you have spatial transcriptomics that is based on spatial sequencing, like spatial barcoding. And they all have their advantages, as you see here. And I really want to quickly kind of like go over this and show you what they all mean. One is spatial resolution. What is the resolution of my picture? Is it at the subcellular level, at the cellular level, maybe at the niche level? And it really matters because if you have a pixelated picture, you can see something. But if you can have really high resolve data, it might look completely different. Then we have the number of molecular targets. Is it complete unbiased? For example, is it poly-A enrichment based, or is it more targeted? Do you need specific targeted probes? Like do you need antibodies to identify proteins or specific gene probes to identify genes? What are the temporal dynamics? Is it live or fixed cells? I can tell you that almost all technologies right now use fixed cells, and that can be snap frozen or formula fixed. Can we assess subcellular function? It can only that is somehow related to the resolution, obviously. What I always think is really interesting to know is that the fluid inside a cell's nucleus is actually 300 times more viscous than honey. So understanding where transcripts originate or where they accumulate can be very informative, and it's not a random process. And then obviously cell throughput, how many cells can be analyzed. It's also very different depending on which technology you use. But typically nowadays, all technologies can easily process hundreds of thousands of cells, and that's usually more than 10 to 100 times more than a single cell RNA-seq data set. All right, so really quickly, spatial proteomics. Spatial proteomics is actually like your immunofluorescence. You have a protein, you have an antibody that recognizes specific antigen from a protein, and that antibody has a fluorophore, so you can measure that. But now you're limited to your fluorophores and secondary antibodies. So a solution is, and there are many solutions, but we're just highlighting one, is different types of multiplexing strategies. And so what Lunaphore does is a sequential IF. So they do IF, and that's this specific part, but then they elute it, and then they kind of like do it over and over again. So it's kind of like repeated or sequential immunofluorescence. And if you do that, there are specific technological innovations that needs to happen in order to make that possible. But eventually, you can analyze multiple different markers at the protein level, all from the same single tissue. You also have like Codex or Akoya, you have IMC, and there's many other technologies that allow you to do something similar, although using slightly different technologies of multiplexing these antibodies. This is just a quick example about how Lunaphore data looks like, and I think we will show one example later on in one of the tutorials about a Lunaphore data set. As I told you before, there are many other spatial proteomics data sets, and I will invite you to read that specific review if you want to know more about how each of them work. Spatial transcriptomics. So spatial transcriptomics that is FISH-based, is as the name says itself, is based on in situ hybridization. So you have oligonucleotide with a fluorophore, but again, you are limited to the number of fluorophores. So what the solution again is the same as we did previously, but now it's like multiplexing these in situ hybridizations. So you want to do this like multiple times. And in this example, I'm giving you an example for seqfish plus, where one transcript, for example, has fluorophore, green fluorophore for transcript 1, you have 1 for transcript 2, it could be red. But now if you start doing this in repeated rounds, so repeated measurements where you wash off and you add new in situ hybridization probes with different colors, you can create this kind of like barcoded schema with different colors that represent different type of transcript, represent different genes. So by doing this with seqfish plus, they showed that you can basically almost capture the entire transcriptome. And this is here an individual single cell in the middle where you can identify the transcripts down to the nanometer kind of like resolution, which I think is really amazing. There are different technologies. We have Merfish, we have seqfish, now Xenium, we have nanostring cosmics. They all do something very similar, and the way they multiplex is slightly different, but nowadays they can easily do this for hundreds, if not thousands of genes all at the same time. Here is some of these examples and how they multiplex it. Again, it's the same really beautiful review that you can read if you want to know more about it. Last but not least, the spatial transcriptomics is probably the most abundantly used. So this is based on in situ capture, and where you try to capture your transcripts using spatial barcodes in one way or the other. The best known is probably Visium, that I think most of you are familiar with, where these barcodes are printed on a special slide from 10x Genomics, and then you pull down your transcripts, they attach, you anneal your barcode to your transcripts, and then you sequence it, so you can both sequence the transcript part, the gene part, and as well as the barcode, so you can push it back to the original locations and you know exactly where each transcript was coming from or where it is expressed. There are definitely many other technologies that you can use, and here are a few of these sequencing-based technologies. All right, so data pre-processing. There's like image processing and then going from raw signal to usable data. I'm going to go to this pretty quickly. So sometimes you cannot analyze your whole image at once, but you have to do multiples, and you have to build multiple fields of view, and then you have to correct for them in stitching. You might have like multiple data sets from adjacent sections, or from the same section, you might have an IF data or an H&E and the spatial proteomics or spatial transcriptomics, which you need to register. So you need to find a way to kind of like get them in the same coordinate framework. Then this is just an example how these images needs to be registered so that they're all nicely aligned. You can make a 3D data set or another type of like registered data set. In addition, there is a specific set of barcoding calling, like how do you exactly call the barcodes? Usually that is provided by the technology itself, like by 10x Genomics, by StereoSeq, or by anything else that you might be using. Segmentation is a very like hot and ongoing topic, like how exactly, if you have single cell information, do you segment your data? It's definitely not a resolved issue. There are many classical approaches that are available through Feature QPAD, and also like a lot of new deep learning approaches that you can apply, and that sometimes work really well and sometimes need a little bit of like up-tuning. Right, the analysis, and you're going to see many of them in the next couple of days. There are different types of downstream analysis, which we have extensively covered in our review from a couple of years ago, that you will see in the next couple of days. And with that, I just want to say, like, this opens up a lot of like new ways of analyzing your favorite tissue or whatever it is, and allows you to do really complex use cases where you can go from both single cell to spatial. But I don't really like this representation. I just want to show you why spatial is so much more than just like what they showed here. It's like RNA-seq, and I think you can envision that as kind of like looking at the ruins of old civilizations, and it's really helpful. I mean, we have learned a lot about the Greeks and the Romans, but if we really want to know how a building is organized, we need to look at the individual constituents. So that would be single cell RNA-seq, and we can learn about what kind of bricks, cement, concrete, everything you want to use. But the functional part really requires you to know the spatial organization. Like the way you build the way you build different houses might give it a very different function. But that's kind of like spatial. But spatial is a lot more because it happens at multiple scales. That house is located in a neighborhood, is located in a bigger city, and the way that city or neighborhood is built might lead to more or less congested roads, make it more amenable for like one thing or the other. In addition, it's easy to generate really pretty pictures from spatial omics technologies, but also to get really insights, you need to make an abstract representation and think about what it really means and what we can learn in the future. And with that one, I just want to show some of these data sets on the future that I call them. These are data sets where they combine multiple spatial technologies from the same section. So oftentimes this is on the same tissue slice, other times it is on adjacent sections. And here is a spatial transcriptomics map where they combine MERFISH with electron microscopy. They can do this spatial epigenome transcriptome. This is from Rong Fan's group. They're also making three-dimensional spatial transcriptomics data set right now. So it's really super cool. And it's really to kind of like allow all of you and all of us to analyze these sort of data sets that we have started building Giotto Suite that makes it a lot easier to work with this like multi-scale and multi-omics technologies. Right, and that's it for this first presentation.